Hello, my name is Mark Stacy. Today is Thursday, October 28th, 2010. It's my pleasure to talk today with Dr. Roger Duvoisin about his neurology career. Roger, I appreciate you doing this, and I think we'll wing this as we go through. I think one of, at least my personal goal, is for people to realize how passionate you are about neurology, how hard you work to make neurology accessible to so many people in the world, and your the influence that your teachers had on you and how you translated that into educating so many other residents and bringing along faculty. I think it's also important that we talk about the major contributions that you work, and I'm always amazed at your bookkeeping of the bookends of levodopa, of proving that levodopa was an actually therapeutic intervention in Parkinson's disease, and then finding the gene for alpha-synuclein. So with all that, I'd like for you to recount really what was the first thing that happened to you to make you think that neurology was something you might want to do?
Well, when I was an intern in 1954 at Lenox Hill Hospital in New York, I didn't know what specialty I wanted to be. I was fascinated by cardiology at the time, especially congenital heart abnormalities, and I was thinking of going into that field. But then I met a neurologist who turned me on to the field. The diagnostic problems were so challenging, and he was very imaginative. And I got to meet a number of neurologists through him. This was an era when arteriography was just beginning. No one knew how to do it at this particular hospital. So I did the first carotid sticks. And lo and behold, we could see the circulation of the living brain. That seemed phenomenal at the time. We could diagnose a subdural or an aneurysm that before we could only guess at. So that turned me on to neurology. And I met one of the attendings at Lenox Hill Hospital was Dr. Melvin Yar, who had been an intern there too some years before, so we had a connection. And he encouraged me to apply to the Neurological Institute to continue my residency in neurology. So I did that, and lo and behold, they accepted me. And I started out as a first-year neurology resident at Columbia Presbyterian. At that time, I was an Air Force resident. The resident salaries were honoraria in those days. I had a wife and 2 children. So as I used to say, I made a contract with the devil like Dr. Faustus. Mine was with the Air Force. They paid me a salary as a first lieutenant. I owed them a year for a year of service, and I did do I owed them 4 years of service. I was a veteran of World War II. I'd been a corpsman in the Navy in World War II. So I did not owe any time on the then Dr. Draft Law. No one today knows about the Dr. Draft Law, but the draft was continued for doctors for a long time. That was not ended until, I guess the 1970s.
That would explain why so many physicians went to Vietnam, I suppose.
Yeah. Well,
so
that's how they got there.
If we get back to Lenox Hill, you told me once that that is where you first met Foster Kennedy. Is that right?
No, he was dead. He died just before I got there. But he had established a neurology service at Lenox Hill. It actually had an approval for 2 years of residency. And I got 1 year of my training there with Dr. Aaron Bell, Dr. Melvin Yar, and other staff members. Dr. Francis Eklund, who was a neurosurgeon. And so I did get some basic neurology experience with those people. But then when I came to the Neurologic Institute, it was a whole different world. He was a hospital, but it was one of the few training programs. I think at that time there were only 15 residencies in the United States. And most of them were in the East Coast, Boston, Philadelphia, and New York.
So Tell us about Houston Merritt. I know you're a great admirer of his.
Well, he was a man of few words. He had a way of having you learn by experience that he supervised. And I'll give you an example. One of my first cases in my first month on duty at the Institute was a young black boy, about 15, whose father was a public health physician. And he had a history of having had a fall down a flight of stairs, and he was carried with the diagnosis of subdural hematoma. They were trying to get a spinal tap on him, and nobody was successful at doing an LP on him for some reason. But I finally succeeded. I was good with a needle, and we got but just before I did the spinal tap, the senior resident was an English fellow, Dr. Michael Small, who was doing a rotation in the States, and he was the senior resident on this ward. So he petitioned for permission to do a cisternal tap on this boy. Dr. Merritt had a rule, no cisternal taps unless he approved of them specifically. So I was called on to present the case to Dr. Merritt. He came down specially for the occasion. I presented the case to him. He looked over at the boy. He took his flashlight out of his pocket, flashed it in the boy's eyes, and he just shook his head no and walked off. Didn't say a word. And we were all upset. John Michael Small was fit to be tied. What were we going to do with it? So I said, well, let me try the spinal tap once more, and then I did it. And we got fluid back, and it came back with positive serology. So then I realized what had happened. He had pupils that did not react to light. Mm-hmm. He had the Ajah Robertson pupil.
Right.
That's what Merritt found with his flashlight. He knew that. That's all he did was take this flashlight. So he
was efficient.
He was right to the diagnosis. That amazed me. So I was then asked to present this case to the students, the clinical clerks session with Dr. Merritt. And I presented the case to the students, and Dr. Merritt then turned to me and said, now do you know why I said no? That was his way of teaching.
Yeah.
And I remember him later on in when I was a senior resident, he said, we had a young black boy, about 17 or 18, who had MS. And Dr. Merritt said, Negroes don't get MS. What? Mm-hmm. So I arranged to present one case each week when a black man with multiple sclerosis. After the third or fourth one, he nodded, he looked at me, nodded, and smiled, and that was the end of that. One of the students was turned on by that. His name escapes me at the moment. He was in Philadelphia. He spent his life on the epidemiology of MS as a result. But that was the teaching by example. He had a reputation of getting the diagnosis right most of the time. And when he couldn't, he would say it was Eastern Siberian camel bite fever, or something like that. And there were times when you just couldn't get a diagnosis.
So if we jump to a little round again, you mentioned the Air Force, and you served in San Antonio after you finished.
After I finished my residency, I served in the Air Force, and I was assigned to the major teaching hospital. The Air Force Medical Service was just developing at that time, and they had this hospital in San Antonio. It was the Lackland Air Force Base Hospital. It became the Wilfred Hall Air Force Hospital, and they planned it as a major teaching center, their equivalent of Walter Reed. And it was during a phase of its expansion that I was there.
One of the fun stories about your life is you took care of a Mercury astronaut who had a syncopal event. Is that right?
Yes. The Air Force was involved with the space program, and they had all the physicians who had any expertise on a panel, and I was on that panel to investigate or evaluate the astronauts, and they were all in hospital for a couple of weeks. We put them through a battery of tests. I was doing a research project at that time on syncope, and I would induce syncope with the Valsalva maneuver, with and without hyperventilation, with an EEG monitor and an EKG monitor ongoing, and I put all the astronauts through that.
So based on your work in the Air Force, you did publish some guidelines, as I remember, about evaluation of astronauts and syncope and what turned into the space evaluation program.
I wrote a few papers on convulsive syncope because there was a discussion at the time about whether convulsive movements that occurred during a faint represented epilepsy or something different. And I argued that it was different. Mm-hmm. First of all, the EEG pattern was that of electrical silence, not an epileptic discharge. Secondly, those were episodes of decerebration.
Right.
And there would be just 4 or 5 jerks usually, and that would be the end of it. And there was a speedy recovery, and there was no post-ictal state.
So would you call that then subcortical myoclonus?
So we call that convulsive syncope.
Yeah, but I think in today's vernacular it would be trying to localize the myoclonic movements. It would have to be a subcortical sort of phenomenon.
Yeah.
Yeah, interesting.
Exactly.
So even when you weren't trying to do movement disorders, you were.
But then when I finished my 4 years at Lackland, and Dr. Merritt offered me a position at Columbia, and so I resigned from the Air Force and I went back to Columbia.
And then when you went back, so we can, since this is a movement disorders tape, maybe we should talk about that. So you went back and you took over a large practice.
Well, what had happened was that the Parkinson's Disease Foundation had been established at Columbia. And Mr. William Black had given the money to build a building, 1 floor of which was to be devoted to Parkinson's research, and the administrative offices of the Parkinson's Disease Foundation that he funded. The personal reason he did that was that his partner and friend of many years had Parkinson's disease. So he put that money to work to research Parkinson's disease. So they established the foundation, they established the Parkinson Center, and I was the first fellow of that organ. They appointed me as a fellow. There were many since then, but I was the first one. And then there was a Parkinson clinic, which dated back to the 1930s. It had been there since the days of encephalitis lethargica. It had originally been established by a grant from Matheson, who had encephalitis himself, and he left this money for research in that field and maintain that clinic. But the clinic, by 1962, when I arrived there from the Air Force, only about 10% of the patients were post-encephalitic. The rest were all Parkinson's disease plus a handful of other things as yet not diagnosed. Some turned out to have progressive supranuclear palsy, some had striatal degeneration, and other things. But the majority had Parkinson's disease.
And when you inherited this big patient population, did you go about trying to catalog them? How did you decide to work with them?
Yes, we did that. We started off cataloging all the patients from 1930 on, did follow-up studies to see what had happened to them, and found that they had a mortality rate, to put it succinctly, about the average patient attended the clinic for about 4 years, and then due to progression of the disease ended up in nursing homes or chronic care facilities at the time. And the death rate was about 3-fold the normal death rate for the population in those years. So we had a background to see what L-DOPA could do.
And that was in about what year? So 62 you started...
Well, that we published in, actually was published by my colleague Margaret Hohen, Peggy Hohen, in 1967, I think, 68. It took some years to do all that analysis. One of the things that we uncovered in our follow-up study was the low incidence of smoking behavior amongst Parkinson patients. There's a group of patients that didn't smoke. And there was a big discussion as to whether they didn't smoke because they had Parkinson's, or whether smoking protected against it, or whether it was an initial symptom that they couldn't get addicted to nicotine.
It would have been helpful to me if you'd have solved that, so I wouldn't have to hear those continuous debates now,
still
40 years later.
The issue's still in the air. I've always thought it had to do with being addictable to nicotine.
Right.
So dopamine is a function. Without dopamine, you don't get addicted.
Right.
What was striking was that about that time, Tardive Dyskinesia appeared. I was called in to look at some of those. I was part of a panel convened by the Institute of Mental Health to look at state hospital populations and look at this new phenomenon. And one thing that struck me was that all the schizophrenics were smoking. High incidence of nicotine.
That's interesting. That's true. Yeah. Some people have published case reports saying that tardive dyskinesia or these involuntary stereotypic movements existed before neuroleptic therapy. Were you ever aware of that?
No. At that time, it seemed a novelty because all of a sudden, 20% of the hospital population had these oral buccal lingual movements, and so it was if there had been some before, there may well have been, but there were cases of Parkinson's disease in the state mental hospital system. And there were other conditions.
So one of the more interesting books I've read recently is that book by Bud Rowland, where he talks about Merritt and Putnam's work in translating diphenylhydantoin from a theory, from a molecule to a patient care, to something that revolutionized epilepsy. Right. I think it would be interesting for you to talk about your experience with levodopa and how, I think someone came to you from Roche maybe to talk about that, but if you'd talk about how that process occurred.
Oh, well, when I came on board in 1962, there already was some talk about levodopa. And it was in 1960 that Hondikiewicz had found a dopamine deficiency in this striatum of Parkinson patients in Vienna. And so for the next 5 or 6 years, many people administered L-Dopa, but usually in very small doses, 25 or 50 or 100 milligrams. And the results were contradictory. There was even a frequently mentioned at that time double-blind study done in Sweden comparing a single dose of L-Dopa to placebo and finding no effect at all. But considering the very low dosages, it's not surprising that they didn't see anything. There was a paper by Patrick McGeer from Vancouver, Canada, and there was a description of about, I think, 5 or 6 cases, all treated with small doses of DL-Dopa. So we later learned that only L-Dopa, only the levo isomer was effective, and the DL was, of course, it took twice the dose.
Right.
And his dosage was, highest dose was, I think, 3 grams a day. And the person who got the highest dose did have some effect, but not much, and also had nausea and concomitant symptoms, side effects. So he concluded that it was useless. But if you looked at his data, the one case that did show some hint of a response was the one with the highest dose. It should have been a clue to, if he had been experienced as a clinical investigator in pharmacology, he would have pursued that. But he didn't. He gave up. The man who pursued it was George Kotzius at Brookhaven, who found that if you pursued giving the L-Dopa, the nausea eventually disappeared, abated. And he could start with low doses and gradually build up over a period of a month. And then he could start to see, when he reached doses of 5 or 6 grams a day of levodopa, then he saw a clinical effect. And that came out in 1967, I think. I remember attending a meeting in Montreal where George presented a film of his patients. And what was striking was that some of the patients showed chorea at the high dose when they were getting a good response to the L-Dopa. That, to me, was significant.
Right.
Because I saw chorea as the opposite of Parkinsonism, as a pathophysiologic state. So that was proof that it was doing something. And I recall the meeting because I jumped up in the discussion period that followed George's paper, I jumped up to congratulate him and point out the career. And then Pat McGee followed me to say he had tried it and it didn't work. And so even then there were people who were not convinced. But we decided at Columbia that we ought to get involved. There were difficulties. The Harris-Kefauver Act had been passed, I think, in 1963. And soon it became impossible to simply take chemicals off the shelf and give them to patients in trials. You had to go through an institutional review board. You had to have adequate testing that said it was safe for human use. You had to be a drug company to have the wherewithal to do that. So we could not use the L-Dopa that was available to us, and the university wouldn't let us administer it. We had a small supply of L-Dopa through a company called Nutritional Biochemicals of Cleveland, as I remember. But we couldn't use it. We couldn't get approval. And then one day, one of the higher executives of Hoffmann La Roche in Switzerland, in Basel, came to see us. He was very interested in the Dopa story. He supplied the L-Dopa to Berkmeyer in Vienna, who was reporting results. And this gentleman wanted to know whether Berkmeyer was a magician or whether this really was effective. And he said he would give us 1 kilogram of L-Dopa and wanted us to try it. So the L-Dopa arrived, and it was promptly impounded at Kennedy Airport because under an Immigration or Tariff Act of 1910 or so, which was designed at the time to protect the then infant American chemical industry, chemicals that were not available in the States, there was a tariff. And the immigration authorities simply looked up a chemical catalog, looked up what the price of L-Dopa was, and it was $25 per milligram. Well, it was used as a laboratory reagent in tiny amounts, so that was reasonable. But if you wanted to give 500 milligrams to a patient, that was too much.
Yeah.
So we had to get the university lawyers involved, and they produced an affidavit to the effect that this was for research. And there was a clause in the law indicating that if it was for research, it was exempt from the tariff. So then they released the material, and we got it. And Hoffmann La Roche packaged it in capsules for us, and then we started our trial. Our first patient started in December '67. By June of '68, or May of '68, we had 38 patients had been treated with dramatic results for most of them. We presented that at a meeting in Scotland in May of '68, and at the American Neurological Association meeting in June '68, Dr. Yar presented that one, and it was sensational. The people in Europe were also using L-Dopa. There was a curious sidelight. The people in Europe knew that there was a lot of dopa decarboxylase in the capillaries of the brain. They thought that L-Dopa couldn't get through that barrier. That was the blood-brain barrier for levodopa. The only area that does not have didn't have the decarboxylase was the area postrema, the vomiting center.
Right.
Which you could understand. Mother Nature arranged things so that we would detect L-Dopa and not eat it. So they had co-administered it with a dopa decarboxylase inhibitor. They didn't realize at first how that worked, and August Pletcher was the higher level executive at Hoffmann La Roche. One of his schemes for testing whether Berkmeyer was a magician or not was to give him some of this dopa decarboxylase inhibitor and told him that it would enhance the effect.
Right.
Or he thought that it would block it.
Right.
Berkmeyer reported that it enhanced it considerably. And he was bamboozled by that, But then they went back to the lab and discovered that it didn't cross the blood-brain barrier.
Right.
So it did not prevent the conversion of dopa to dopamine in the brain once it got in.
Right.
And it broke down the blood-brain barrier, so it got in.
Interesting.
So then that became, their trademark name for it was Madopar, which as far as I know is still used everywhere in the world except the United States.
Right.
But at that time, the Hoppen LaRoche had invested a considerable amount of money to develop an L-Dopa plant. They, as I understood, took over part of their vitamin C factory and converted it to Dopa. They made it from vanilla, and they had L-Dopa from L-vanilla. Excuse me. As I recall, the Hoppen LaRoche produced levodopa under the trademark name Larodopa. And then Merck Sharp and Dome produced another dopa decarboxylase inhibitor, Carbidopa, which was more potent than the Hoppen LaRoche drug. And they packaged it in combination with levodopa. Now, levodopa was not patentable. It was a product of nature. But Carbidopa was. And that put Hoppen LaRoche in a bind. They could still produce Larodopa, but they couldn't produce Cinemet.
Right.
Merck Sharp and Dome cleverly gave it the name Cinemet, meaning in Latin, without vomiting.
It's a good name.
And indeed, that was its major benefit, is that you could give small doses.
Right.
You could get up to a therapeutic dose very quickly. And there you were. Now the question was, had you cured Parkinson's disease? Or had you simply relieved the symptoms but the disease progressed? That was a big discussion. Or had the presence of levodopa accelerated the disease? Always a historic physician fear that of treatment. It's like whipping a tired horse. Analogy is going to make the disease ultimately worse. And that was a hot topic of discussion. Anyway, by 1973, my term at Columbia Presbyterian, I took a sabbatical and went to London for 6 months. And I worked with David Marsden in what was it was south of the Thames.
Queen Square.
Not Queen Square, no.
Was it King's College then?
King's College. King's College Hospital Medical School. And affiliated to it was a psychiatric hospital, the Royal Bethlehem Hospital of Bedlam fame, which is a big psychiatric center. But anyway, I worked there for 6 months on animal models of Parkinson's and learned how to make a stereotaxic lesion in rats. And I came home and continued that work. But I came home in 1973, and my senior associate, Dr. Melvin Yar, had accepted the position of chairman of neurology at Mount Sinai, which was then a new medical school. And so I accompanied him there and became a professor there. I stayed there for 6 years. By this time, it was L-Dopa. L-Dopa was approved by the FDA in 1970, and Carbidopa within a couple of years of that. And there didn't seem to be anything more to do. And now, so many years later, levodopa is still the major treatment for Parkinson's disease. It's well established now, I think, that it does not affect the course of the disease. It's simply symptomatic relief, for which we're very grateful. To some patients, it's a lifesaver, and it has extended the life expectancy. The last I have looked at the figures, it appears that the life expectancy of Parkinson's disease patient approximates the normal, or comes quite close to it, which is a great improvement over what it was like before. Now, there are lots of other things that have happened. In 1965, at the American Neurological Association meeting, an eminent Canadian neurologist named Richardson presented several cases of a newly discovered disorder. Actually, it had been described before under various names, but no one had put it together. He had 6 or 7 cases. They called it heterogeneous system degeneration, I think. It looked like Parkinsonism, but not quite. The pathologist in that case, Olszewski, when he first did an autopsy on one of these patients, thought it was post-encephalitic Parkinsonism. And Richardson told him, no, it can't be. It's something else. Go back and check again, so, which he did do. Now, what led him astray was that there were neurofibrillary tangles but the distribution of the lesions was different, and he realized that, oh, this was a different disorder. It later became called progressive supranuclear palsy. One of Richardson's residents was John Steele, who wrote up the papers, and it's known today as the Steele-Richardson-Olszewski syndrome, or PSP for short. Well, I remember seeing the very first case that we had at Columbia Presbyterian. Dr. Merritt, we presented it to him. He said I remember him standing at the bedside, saying he had never seen that before. And I asked him, well, where were they before? What were they called? And he said, well, they probably were called post-encephalitic, which is probably true.
Right.
In the early 60s, when I started out at Columbia in 62, I was in charge of collecting the clinical data for the Parkinson Brain Bank that the Parkinson Foundation had established. And the neuropathologist at Columbia, Dr. Abner Wolf, was in charge of the pathologic investigation of these brains. And he assigned a fellow, Manny Stadlin, to work with me on these cases. So we did an analysis of the first 25 or so cases, nearly all of which had been patients. So we had clinical records on them from Columbia. It turned out that there were, as I recall now, there were 2 cases of post-encephalitic Parkinsonism in the group. There were 1 or 2 cases who did not have Lewy bodies, and we thought that they corresponded to a condition that had just been reported by Adams as striatonigual degeneration, which Adams claimed clinicians couldn't distinguish from Parkinson's disease. But it wasn't Parkinson's disease. It had a different pathology. It was in the striatum. So there were a couple of those. And there was another case that we couldn't decide what it was. And that was it. So the Lewy body was important. Most of the patients had Lewy body. And what was the Lewy body? Big discussion as to the Lewy body and its construction and what did it consist of. The answer to that did not come until after we found the gene.
So let's talk about the gene. You always were a proponent that there had to be some genetic component to Parkinson's disease, and you told me that was from going to support groups. But if you could talk about the story of the gene, it would be interesting.
Yeah, when I came to Columbia in 62, Dr. Yar had the idea that we should look into the search for familial cases. He had an idea that maybe there was a genetic factor. And part of the idea came from a published Swedish study by Henry Jones, M-J-O-N-E-S, a Swedish psychiatrist who did a study for his MD, or a graduate degree thesis in Sweden. And he did a study of Parkinson's disease patients. Now, he did not have the advantage of post-mortem confirmation of any. They didn't even agree on what the pathology was at that time. Mm-hmm. And he claimed that it was an autosomal dominant disorder with a low penetrance. and a partial penetrance, normal age range, normal life expectancy. So the question was this true or was it not? And he had no controls. So he had engaged a gentleman, a professor of epidemiology from the School of Public Health, to set up a study of the familial occurrence of Parkinson's disease with controls. So we used the spouses as controls. We identified patients in the clinic, and we spent about four years on this study. We identified patients. We then, a team of us, Dr. Hohen, Dr. Barrett, myself, Dr. Shear, and Dr. Yar, we examined all the siblings of the patients, their spouses and all the spouse siblings that we could get our hands on. And after 4 years, we had, I think, 2 or 3 affected siblings and 2 affected spouse siblings. And that was not a significant difference for the numbers involved, so we could not confirm a familial concentration of Parkinson's disease. But that left me uncertain what was going on here. In the meantime, I had collected several cases of identical twins. I had 3 patients in the clinic who were members of identical twin pairs, and the co-twin was unaffected in all 3 cases. I talked with Dr. Schwab in Boston, who also had 3 cases, and we were going to publish that, but he died and then came to know it. So I continued the study of twins. I started collecting twins. It eventually became, I continued it when I moved to Mount Sinai, and it eventually became a collaborative study with Dr. Cal at the National Institutes of Health. And eventually we studied 65 pairs of twins, identical twins. And we came up with 0 difference. So that was strong evidence that there was not a genetic factor in Parkinson's disease. But in the meantime, I had begun noticing in the patients that I was following that the siblings, I was beginning to see affected siblings. And I picked up cases where a parent was affected and a child was affected. And as the years went by, I saw more and more of these. And when the L-DOPA story came in 1968, I was invited to talk to Parkinson support groups. That seemed to give a big stimulus, by the way, the L-DOPA story. The Parkinson support groups developed all over the country. So I went and gave talks. And I talked about the genetics and said it wasn't genetic. And I remember giving a talk in Ohio, in Columbus, Ohio, and I said it was not genetic. And the man in the audience says, yes, but it's in my family. My uncle, my grandfather, my father had it. So you must have something else. But he looked like real Parkinson's, I must admit. Well, that set me to thinking, and I asked the audience, well, how many of you have an affected relative? And it turns out that about 1/3 of the audience raised their hands. And then I started taking genealogical histories of these people. And I did this all over the country. Seattle, Vancouver, San Antonio, everywhere was the same. So I began to think that there was something to this. And by this time, I was at Rutgers Medical. After 6 years at Mount Sinai, I accepted the position of chairman of a new department at Rutgers Medical School in New Brunswick, New Jersey. It's now called the Robert Wood Johnson Medical School. And I asked one of my colleagues, Larry Golby, if he would do genealogical histories on all our patients. And he did. And he uncovered a family. And it's interesting that one of the members of this family died. He died accidentally in his swimming pool. And it was a medical examiner's case who allowed our pathologist to do the autopsy, and it was Parkinson's disease with Lewy bodies. So we had autopsy confirmation that the familial disease in that family was Lewy body Parkinson's disease. It turned out to be a huge pedigree. And there were actually 2 related families. And they all were descendants of 2 immigrants who came from southern Italy. They all came from a little village on the southern slope of Mount Vesuvius. We had a collaboration with the Department of Neurology at the University of Naples, and I sent Larry Golby there, who went with by this time I had a clinical geneticist working with us. And we collected this pedigree and we published it. It was the first published family in which there was autopsy confirmation of the diagnosis of Lewy body disease. And then I was approached by the director of the NIH. Would I make these families available to their genetic research group? So we agreed to do that. It was a collaboration. And as luck would have it, the young geneticist, he was actually a psychiatrist, Paula discovered the gene within a few weeks on chromosome 4. Shortly after that, that turned out to be a gene for the what is it? Alpha-synuclein. An English neuropathologist showed that that was what the Lewy body consisted of by immunohistochemistry. So we'd solved the riddle of the Lewy body. It was a collection of an abnormal protein. It meant that a number of diseases were, like Alzheimer's, had a collection of abnormal proteins in nerve cells. So the idea was, well, if you could prevent the accumulation,
Right.
you could prevent the disease. Well, there was a great deal of hope at first. So far, no one has found
Less
a
hope.
effective way of doing that.
You want to take a little break?
Yes.
Then I'll look at, I'm supposed to ask you specific questions, but I think your answers are more interesting than their questions.
Yeah, they're just guidelines.
All right.
Do you want
Okay.
some water, Dr. Stacy?
Sure. Although I'm not doing much talking. Are you doing all right with it? Am I wearing you out?
No, that's fine.
Okay.
I hope I'm not boring you.
Well, I'm not wearing me, and I'm the person asking the question, so. Why don't we ask maybe more difficult questions for a modest man like you. Tell me what you believe your greatest contribution was to the field of neurology.
Oh, wow.
I knew that would bother you.
Well, there was several contributions that I take some pride in. One was the business about the convulsive syncope, that the convulsive movements accompanying a faint were not did not represent epilepsy. That at the time was important. Nowadays, it's nothing.
Right.
Then I think shortly after I arrived at Columbia in '62, took over the Parkinson Clinic and started looking at the patients, there was a lot of discussion about the possibility that all Parkinsonism was post-encephalitic. There was a group at Harvard that claimed that argument. And Dr. Merritt had asked me to comment on it, which I did do. Fortunately, there were records. There had been a neurology clinic at Columbia established in 1878, I think. And someone published a survey of all the patients seen in that clinic as of 1903. So that was over 25 years of follow-up, and the number of patients was quite large. And they looked just like the patients that I was seeing in 1962, so far as the descriptions went. There was really no difference. The age of onsets were the same. The symptoms were the same. And so we had a big dispute. It ended up in a couple of papers challenging the thesis that all Parkinson's was post-encephalitic. The chaps at David Postganzer was the young fellow at Harvard who had that thesis, and he based his argument on age of onset. And he looked at the age of onset of all the Parkinson patients in the clinic records that he had unearthed at Harvard, at the Mass General Hospital. What he didn't realize was that in 1920 the hospital began publishing the outpatient statistics in a separate volume from the inpatient statistics. Prior to that, they were all in one volume.
Right.
So the total number of patients seen prior to 1920 was quite large. Then from 1920 on, the hospital had only the very few people who were admitted for Parkinson's disease. The number fell. And the ones that were admitted were all post-encephalitic. So that threw him off, and he didn't recognize that. The other thing that intrigued me was there was a question about how drugs worked. There was a bevy of anticholinergic agents available in those days, Artane, Pagitane, Chemedrine, or Cogentin. And there were claims that one was better for rigidity and one better for tremor, and they had different effects. So I challenged that, and I found that if one administered physostigmine to a Parkinson's patient, you made him abruptly worse, and you canceled the benefit of any anticholinergic drug. If you then gave him an anticholinergic, you reversed the effect, and you could play that back and forth. So there was an antagonism between physostigmine and the anticholinergics. So there was, that indicated there was an imbalance, and the suggestion was the imbalance was between acetylcholine and dopamine. Dopamine was deficient, acetylcholine was predominant. If you blocked the acetylcholine, you could restore the balance somewhat. That was our theory. That was my thesis for my membership to the ANA. In those days, you had to submit a thesis to become a member of the ANA, the American Neurological Association.
That's a pretty good thesis.
So I did that. There was it did draw some, I think that was an important contribution at the time. I felt rather pleased with that. But it didn't get much attention. It got some. It was a clinical pharmacologic experiment. Acetylcholine was then, I think, the only neurotransmitter known in the brain that had been identified. That's dopamine that was then discovered. And I think the major thing I take credit for was recognizing the familial concentration of the disease. And that meant a big change of opinion. I had been touting that it was not because of the twin study results. And then I realized that the twin study was not adequate. And the follow-up on the twin study, a couple of the co-twins became affected later, but much later, and that was troublesome. One of them became affected 26 years after the first one. Why would two identical twins develop Parkinson's disease, but one 26 years after the other? There was some other factor must be involved, of course. And one of the interesting things I learned about identical twins is that there are different kinds of identical twins. Many of them are mirror images of each other. One is left-handed and one is right-handed, for example, but not always. And their teeth may be somewhat different. They may have minor differences. And one has to follow them for a much longer period of time, for a lifetime. Right. To draw any conclusions. So that's why one had to go back to family. And then in the 1970s, with the advent of DNA technology, looking at identical twins to answer questions like whether something was genetic or not was superseded. It was easier to look for a gene in a family. Right. So there was an incentive to collect families so that DNA analysis could be undertaken.
What are your regrets of your career? What would you want to do over?
Oh, that's a tough one. I don't know. I remember when I was an intern and then a medical resident at Lenox Hill. And, I could have gone in so many different directions at that point in time. It's like life is a hallway. You're going down and you pass these hallways, and you close these doors and you close these opportunities. What if you had taken one of the other hallways? What would have happened?
One time you told me that you regretted you could never become a neuropathologist because of levodopa getting in the way.
Yes, when I was struggling with trying to get at the basic nature of Parkinson's disease, the approach of neuropathology appealed to me. And I was on the verge of going into neuropathology with Abner Wolf. But when the L-DOPA story came along, we needed all the manpower we could get. Just didn't have the time anymore.
Right.
It was, couldn't do all of that. Yeah, I could have done neuropathology. Earlier on, when I was in the Air Force working on the question of epilepsy, that too was a field that I might have been attracted to. And I was interested in electroencephalography. I could have pursued that, but I didn't. I was on the verge of trying to qualify for the in those days there was no EEG board as such. Then the criteria began to evolve for recognizing training in that field, and I'd have to get some.
What advice would you give for somebody just finishing a neurology training, just starting their career?
Oh, that's a toughie. Well, one of the things that intrigued me was that being I was in an academic institution, I did not have laboratory skills, and that was a how shall I say? It was a negative. If you wanted academic promotion, you had to have a laboratory. You had to get an NIH grant. And in fact, you couldn't get tenure unless you had an NIH grant. But I was insisting on the clinical skills. I thought clinical skills were dying, that they were important to preserve. And I remember Bud Rowland telling me, well, the clinicians have described everything. Now it's up to the chemist to find the cause. And he spent his lifetime trying to find the cause of muscular dystrophy. The answer did not come from chemistry.
Right.
In the meantime, a lot of advances in the clinical side occurred in Parkinson's. The discovery of the various Parkinson plus syndromes, of which there are now many. That was not from chemistry, that was from clinical and pathology. So there still is an importance to good, sound clinical neurology. It's hard to get credit for it, but it's still very important. And it's a shame to see the skills being lost. The old-timers, I remember, did not have all the wonderful diagnostic aids. They didn't have MRIs or CAT scans or arteriograms. And I saw the effect of the advent of these technologies. They did advance neurology, but they didn't replace it. In fact, they supplemented it. And you still need the clinical skills to combine with these additional new technologies. What new technologies will appear, I don't know, but there'll be some.
When I was a resident, Dr. Mancall, that's who I trained with, told me one time when we were on the floors that he was a heck of a neurologist before that CT scan was invented. I never ever saw him once need to have anything but verification of exactly what he said the lesion where it was by his CT scan, but I always thought that was a very funny remark. And we depend a lot more on a CT scan than he did.
Oh, yeah. I mean, people like Foster Kennedy, supposedly, you can point to the head and tell the surgeon, this is where it is.
Tell me, in your, you mentioned Houston Merritt being a parsimonious but a great neurologist. Tell me, in your experience, who were the neurologists that you thought were your best clinical teachers?
Well, one was my senior partner, Melvin Yar. I worked with him for many years. He was an astute clinician. He didn't get the recognition he deserved as a very sharp clinician. There were the people had their qualities and their faults. amongst the staff at the Neurologic Institute, when I was there was a German immigrant named Ernst Hertz, who had pioneered movement disorders in Germany in the 20s. He made films. He had a Film of the Month Club.
Mm-hmm.
And he would show movement disorders. He had a collection of films. I saw him as an older man who had set up a photo lab of photography, a room devoted to the photography, cinematography of movement disorders, which he did at Columbia. And I used the same room to do the
Mm-hmm.
films on the L-Dopa cases. So I learned a lot from him. Some had very different opinions. I remember Louis Daucher was an old-time practitioner. He knew his brain anatomy extremely well. He was a good teacher of anatomy. But his ideas about Parkinson's disease were off the wall. What could I say? And he had worked with an anatomist with a machine that recorded tremor, and he was studying tremor in patients, and people had tremor recorded, and he was sure that studying that tremor would reveal something.
Mm-hmm.
I thought that was not going to show anything.
So you mentioned Melvin Yar. I do think that he doesn't get near the credit that he should get. He was a man that had strong opinions, and maybe that was part of it. But tell me the essence of him.
Well, he was a tough guy from the Bronx. He grew up in the city. He worked his way through college, NYU. Then he went to NYU Medical School. And he became an intern at Lenox Hill. And then the Army drafted him in World War II. He was in the Army for several years. He was assigned to neurology by the Army. And he liked it. It stuck with him. And when he came out, he became a resident with Houston Merritt at the Montefiore Hospital in New York. The old Montefiore Hospital was an old training center for American neurologists back to the 19th century. It was a chronic hospital. It was a museum. I remember we used to have patients with dystonia musculorum deformans, who were long-time residents of the Montefiore Hospital. We would bring them to the Neurologic Institute with a board exam,
Mm-hmm.
which was held at the Institute. Or cases of Wilson's disease. If you wanted them, you went to Montefiore to find them.
Mm-hmm.
So that's why Merritt went there when he left Boston on his way to Columbia. I think there was an old-time clinician who was a very astute observer of gaits. His name was Carmen Vicali. He was trained at the Neurologic Institute. He was a private practitioner. He didn't publish anything, but he lectured the students. He was a very sharp diagnostician. And I learned some things from him. I mean, different people taught me different things. Right. On the other hand, double-gated molecules, as he called them, didn't impress him. He had no feeling for that, for the scientific basis of neurology. He taught 1901 physiology.
But he knew it.
But he knew it,
Yeah.
And it had a simple explanation.
Right. Tell me what you enjoyed most about your career.
It was my first year as a resident at the Neurologic Institute, and turned me on. Well, I was already turned on, but just to be there was exciting at that time, with a community of people thinking along the same line and concerned about the nature of these things we were seeing. It was heady. I remember when I finished my residency and I came back 6 months later one day to visit. It was a whole different place. I didn't fit in anymore. Nobody, there was a whole new crew. You realize that institutions had lives of their own.
Right.
And you come and go.
Very fast generations.
That's right.
Speaking of changing neurology, how do you think neurology has changed from your days really at probably at Robert Wood Johnson to today?
I mean, since I retired. At first, it seemed to me that I kept seeing the wheel being reinvented all the time. But it is changing in subtle ways. The importance of genetics is hard to underestimate. Should not be underestimated. It's enormous. You see the number of articles that relate to genetics or have genetic technology involved. It's almost half the articles published in neurology now, which it's very hard for me to follow. I am not trained in that field. I think that makes it very difficult for a clinician. There's a technology out there that is all-absorbing. The number of diagnostic entities has grown tremendously. I don't know how anybody can keep track. So it means subspecialization is inevitable. In the 1960s, there were very few pediatric neurologists. There weren't many neurologists in the country. There were a lot of neuropsychiatrists. But as Dr. Merritt used to say, they were people who made their living in psychiatry and got their pleasure from neurology. but they didn't know much neurology either. And they've been replaced. Most of the academic neurologists of my generation came out of internal medicine. And I think the growth of the National Institute of Neurological Disease had a lot to do with that. Now it's a smaller and smaller part of the pie. Child neurology, my gosh, there was 2 or 3 kinds of muscular dystrophy. Now there are, I don't know, 50.
Right.
How do you recognize them? You recognize conditions of which there are only 10 cases in the world. So you recognize conditions that the average practitioner never sees or sees once in a lifetime.
Right. So now, how long have you been retired?
I retired in oh my goodness, 1997.
So what kind of things do you do now, 13 years later?
Well, I still read neurology. I get the journal. I read less and less now than I did. I've been involved with a colleague at University of Indiana writing on encephalitis lethargica. I was busy translating a French document, the MD thesis of Tretiakov on Parkinson's disease, published in 1917. It was the first confirmation of that the substantia nigra was the pathological site for Parkinson's disease, and documentation of the Lewy body. Confirmation of the Lewy body. It was part of a long dialogue. The debate amongst pathologists lasted for a good half-century. It wasn't until the 1960s that they generally agreed that A, the nigra was the site of the pathology, and B, Lewy body was the hallmark. And now, of course, you can have Parkinson's disease without the Lewy bodies, or at least syndromes that you can't clinically distinguish. And now, of course, the Lewy body is much more extensive than it was when you were just looking at it under H&E stains. How else has neurology changed? Well, maybe you can tell me better than I can.
Oh, this is your interview, so I'm not going to say a word. But I do appreciate you coming in and sharing some thoughts. Do you have any other things you want to, you're dying to share?
Oh, well, I would hope that the clinical skills would be encouraged and developed and maintained, and not lost in the technology.
If you got energy for one more, to me, I came to movement disorders just as the Movement Disorder Society was being founded. So the first meeting in Washington, D.C., I remember Mark Hallett trying to look at myoclonus and we voting on what
yes.
myoclonus was about.
I remember that.
And I remember David Marsden really taking over the session one time, being so excited about something that was presented. Since Mark trained with Dr. Marsden, and you spent some time with Dr. Marsden also, to me, he felt like the greatest clinician I'd ever seen. Was that true, or was that just the easy impressionability of a young man starting in movement disorders?
Oh, he was a good clinician. And he saw a wealth of material, enormous wealth. The way the English system was structured, he was sitting on top of a pile of, and he could see this huge volume of clinical material. Yeah, he was very good, very sharp. And he had guts. He had the nerve to challenge authority, and I found him great fun to work with.
So Peter Jenner says that they would go out on Fridays and they would listen to him talk, and and Peter would take notes on the coasters in the bar. And the next morning he would try to decipher the coasters. And that's how things were driven. Is that true?
Could be. I'd never heard that before, but yeah, I remember Peter Jenner. I did 6 months, 7 months actually, with David Marsden, and we had a great time. There were other great neurologists too. I mean, some of the top English neurologists are quite outstanding. There's something about their system that leaves you a top layer of very experienced people who get to see a tremendous amount of material. I remember people saying, well, if it wasn't in Wilson's textbook, it didn't exist.
Right. So MacDonald Critchley wrote a book a long time ago, The Divine Banquet of the Brain.
Yes.
And he talked about his friends and people he knew. Who would be in your book of The Divine Banquet of the Brain for movement disorders?
Oh, my goodness. Well, it's interesting. There are those who organized meetings and conferences and did things that way. That I would place Stanley Fawn there. And he used the Parkinsons Disease Foundation's resources quite thoughtfully. The development of that little session that he and David Marsden used to have at the academy meetings.
Right.
People would show videotapes of things. It was quite instructive. Who else would I have in the department? Well, because some are deceased now. Yeah, there was Harold Clowans, who was quite funny, but yet he became more interested in being a writer than a clinician, and gave up the clinical part. Chris Getz was a very good teacher. He has a special interest in 19th century French neurology, so he writes about Charcot. But I've had Chris as a visiting professor, and he was really a very good teacher with the students. I had Donald Cowan, made important contributions. We disagreed generally. We were on opposite sides of the genetics question for a long time, and we switched. He was for, then he was against, and vice versa. There were some people who I liked Carly Tanner, who tried to make study epidemiology of neurological disorders. She'd had a hard time with the twin study. That was a serious attempt to try, but I don't think it was productive. The discovery of these genes. There are others, but I don't know them personally too well. There was Yves Agide in Paris, who decided to learn neurochemistry. He was going to solve problems in the brain that way, doing the chemistry. French neurology was not very strong on basic science, and he was trying to reverse that. Oh, there are a couple of Italian neurologists that I've met who were quite good, and I don't remember their names now. In Japan, there was Narabayashi, who was a neurologist,
Right.
but who did stereotaxic surgery.
Right.
And he had some interesting ideas about the nature of tremor. But he's gone now. There was, there was Denny Brown, I mentioned, but actually I think Ray Adams was the consummate neurologist, neuropathologist. And he spawned a lot of trainees. I had an interesting experience with Ray Adams. I was invited once to be a visiting professor at Harvard. I went up to give a talk, and I asked him about Denny Brown. Denny Brown's little book on the basal ganglia. He pays no attention to the Lewy body or the substantia nigra.
Mm-hmm.
And I asked Adams why that was so, and he said to me, "Oh, well, Denny was not a good pathologist." He said there was a portrait. Denny Brown was recently deceased. This portrait was hanging on the wall of the Harvard Club. Wow. And I gave my talk the next day. It was a talk about MPTP, and I had shown that it destroyed the substantia nigra in the mouse, and I showed slides of the mouse substantia nigra being wiped out and the behavior of the mice. And at the end of my talk, Adams got up and said, "That case of Denny Brown's from his book, well, I went and cut some more sections, and here are the Lewy bodies."
Overnight.
Yeah, they're all saved up, ready to go. So it's interesting. Well, why did Denny Brown ignore that?
Right.
When I gave the talk at the meeting in Edinburgh, Scotland, in May of '68, and it's happened that Denny Brown chaired the session in which I presented our films, our patients from Columbia. And they showed the movement disorders, they showed the chorea and the relief of the Parkinsons. And I asked him whether he had any explanation for the reverse relationship, the inverse relationship of chorea and Parkinsonism. And he looked at me and he smiled and he said, "You know, since I don't believe the substantia nigra is the site of pathology of Parkinson's, I can't answer your question." Which I took to mean that he was beginning to think about it.
Yeah. Well, it's unanswerable if you can't accept that.
Yeah.
Yeah.
So he wasn't quite sure about that. But I took it to mean that he was thinking about it.
Yeah.
He probably would have come around.
With the few minutes we have left, can you give advice to young people regarding on how you dealt with barriers, if they were put in your way, how you were able to remain with your basic decency and kindness as I talk to you today, all the way through a career that is remarkably successful? There must have been barriers and potential fights that were put in your way. How did you avoid them or negotiate them?
That's a tough question. There was one barrier, of course, was the fact that I did not have a laboratory experience. I had no pharmacology or biochemistry or etc. And to say that I was a clinician and my patients were my lab rats was difficult to convey. But I was making progress. It's harder to work with patients, with human beings, than with animals. And the answers that you get from the animals are not necessarily quite as informative as you may think they are. Something seems to work in the animals and it doesn't work in the human. So I think that was one barrier that I faced, that I did not have the laboratory background. Still, you can't do everything. I think the importance of the laboratory background was greater in the 60s than it is today. Whether it's a recognition of that or the greater difficulty of achieving more PhDs rather than MDs are getting laboratory research posts at the NIH, for example. And that's okay, that's fine. That may be perfectly all right because life is short. What was that? As Hippocrates put it. Art is long and life is short, and experience is misleading. And so true. I think my advice would be find something that really engages you and devote yourself to it. And put the worries about finances on the back burner. Because in the long run, you will wonder whether your decisions you made are worthwhile. What struck me about my colleagues who went into clinical practice to make ends meet is that they got bored. They couldn't go to the meetings because they couldn't afford the time away from their practice. So they lost the intellectual stimulus was gone. And finally, they were left behind. And the conversation that you have with your colleagues in the scientific field worldwide is something to treasure. That's part of my life experience. I think that I enjoyed the most was my relationship with neurologists in Europe and South America and Japan. And the battle of ideas. If only you can continue them. And you may have to reach their specialization is such that you may only find partners in conversation in other parts of the world, far away from where you are. And yet you may find people who have identical situations with yours. I think medicine is an international profession more than any other. It's one thing to be a lawyer, or I've got two daughters who are lawyers, but they don't have any foreign legal experience or exposure, whereas I had a lot of foreign exposure in my field. I think that would be my closing thought.
Thank you for your time this afternoon.
Okay, my pleasure.
Thanks so much.